When we search for the origins of life, we often look for a spark, a moment where a self-contained creature emerges from a primordial soup. But the modern chemical evidence suggests that the very first living things remained physically tethered to the planet's geology. All life on Earth traces back to a single common ancestor, known as LUCA. By mapping the genetic legacy of LUCA, scientists have reconstructed its internal metabolic network. What they found is a system that looks unfinished. It is a map defined by massive structural gaps where essential chemical pathways should be. Specifically, LUCA lacked the enzymes needed for protein synthesis. It was missing the biological tools required to create amino acids, the very components it needed to stay alive. A biological machine cannot function with a broken engine. Without a way to manufacture its own building blocks, LUCA would have been unable to exist as a free-swimming cell. This incomplete state indicated that LUCA relied on its environment to complete its chemical circuits. It survived by using the surrounding seafloor as an external life support system. On the Hadean Earth, the deep ocean floor was dominated by serpentinizing hydrothermal vents. These towering mineral chimneys pumped hot, alkaline fluid into the seawater. These vents provided the physical scaffolding for early metabolism, acting as the external source for LUCA's missing biological components. Within the rock itself, deposits of transition metals, specifically nickel, iron, and cobalt, served as the forerunners to biological enzymes. This geological catalysis drove the acetyl-CoA pathway. The metal surfaces grabbed dissolved carbon dioxide and hydrogen from the vent fluid, stripping away their electrons and forcing them to bond into the organic precursors of life. At this stage, the metabolism of life and the geology of the planet were the same thing. The crust of the Earth performed the digestion and synthesis that the proto-organism could not. Building organic matter requires more than just a catalyst. It needs a continuous supply of chemical energy to drive the reactions forward. This energy came from phosphite, a highly reactive form of phosphorus found in the surrounding minerals. When phosphite reacted with palladium, it triggered an oxidation reaction, forging high-energy phosphate bonds. These bonds provided a primordial battery. By slotting into the gaps of LUCA's incomplete metabolic network, this geological energy allowed the biological system to function. With the vent providing both the catalysts and the fuel, the living system remained physically inseparable from the rock. If the earliest stages of life were bound to the seafloor, we have to ask how biology ever detached to become a mobile, independent cell. Over vast spans of time, the metabolic network began synthesizing its own organic molecules to take over the work of the rock. These molecules, called cofactors, were designed to mimic the metals in the crust. As shown here, when a cofactor enters the network, it replaces the immobile mineral node with a mobile biological equivalent. This transition created an autocatalytic state. Life began manufacturing its own catalysts, providing the independence needed to survive away from the mineral surfaces. The final step was encapsulation. The system used lipids to package these reactions within a microscopic membrane, allowing the chemistry to leave the rock. The first cells survived by carrying the vent's specific chemical conditions with them. They portabilized the catalytic power of the crust, enabling the processes of the seafloor to persist in the open ocean. When we look for life elsewhere in the universe, we are looking for this specific dialogue between biology and geology. We are searching for a system that begins not as a swimmer, but as a living extension of an active planetary crust. 4 billion years ago, the bottom of the ocean was a site of constant chemical activity. Scalding hydrothermal vents were blasting highly alkaline fluids out of cracks in the crust, directly into the freezing, dark seawater above. But if you zoom in on the jagged surfaces of these vents, you'll find they are coated in transition metals, elements like iron, nickel, and cobalt, sitting directly exposed in the rock. These metals are highly reactive. In this deep sea environment, they physically grab carbon dioxide from the water, force it to react, and stitch together amino acids, the specific organic building blocks that make up cellular proteins. Long before a single biological cell existed, the Earth's crust was acting as a chemical engine. The rocks themselves were performing the complex synthesis we normally assume requires a living organism. We can trace the genetics of modern organisms back to a theoretical starting point, LUCA, the last universal common ancestor. But mapping LUCA's genetics reveals a structural disconnect. Its internal metabolism was incomplete. This diagram shows LUCA's metabolic network, a chemical circuit for processing nutrients. But notice where the network stops. It lacks the biological enzymes to synthesize essential amino acids. A cell needs 400 continuous reactions to survive. When pathways sever, the circuit fails completely. LUCA lacked the internal machinery to survive on its own. It depended on the chemistry provided by its outside environment to bridge the gaps in its reactions. To figure out how this early organism stayed alive, researchers analyzed the specific chemical reactions that LUCA couldn't perform itself. They discovered a match. The exact reactions missing from LUCA's biology are driven by the transition metals found on hydrothermal vents. Iron, nickel, and cobalt slot directly into those empty spaces. These environmental metals acted as the original catalysts. They performed the exact structural and functional roles that biological enzymes handle today, completing the circuit so the cell could function. Early life didn't invent a novel set of chemical rules. Biology simply adopted the chemical reactions the deep-sea rocks were already performing and built a cell around them. Connecting the pathways is only half the problem. Chemical reactions require a continuous energy source to drive them forward. In modern cells, this process, known as phosphorylation, relies on a specialized biological battery called ATP. Early organisms didn't have the biological machinery to produce their own high-energy bonds. Without a way to power those reactions, survival was thermodynamically impossible. The minerals within the vent environment activated this process. Elements like palladium and nickel, embedded in the rock, naturally interact with an environmental compound called phosphite. This interaction strips away electrons and generates life's energy currency entirely without biological enzymes. The hydrothermal vent functioned as a continuous thermodynamic battery, driving life's first chemical steps in the safety of its rocky chambers. If the deep-sea rocks were doing all this chemical work effectively, we have to ask why life bothered to evolve its own biological enzymes at all. Relying on a geological scaffold comes with a severe limitation. Rock surfaces are completely stationary, and they lack precise chemical specificity. As long as an organism relied on the vent to complete its metabolism, it was physically chained to the rock. To solve this, early metabolism synthesized cofactors, complex organic molecules replicating the exact catalytic work of vent metals. Serving as miniaturized, mobile versions, they plugged directly into the cell's biological structures, allowing early life to finally gain mobility. The ability to synthesize organic cofactors made early metabolism autocatalytic. The organism could finally sustain its own chemical reactions without relying on a piece of the ocean floor. A moment of wonder. Early cellular structures could finally detach from the jagged rock surfaces that birthed them, drifting upward into the open ocean to survive on their own. The origin of life occurred through a slow physical transfer of power. Biology encapsulated the chemistry of the primordial vent inside a membrane, internalizing the Earth's geology so it could finally swim away. Current scientific reconstructions of LUCA, the last universal common ancestor, produce an impossible contradiction. The methodologies used to find our ancestor yield results that cannot exist in the same cell. This 2024 phylogenetic gene tree depicts LUCA as a highly complex organism, but on the right, a map of the metabolic core from 953 living prokaryotes shows a different result. Mapping enzymes required for basic amino acids reveals severe gaps. The pathways are incomplete. A cell cannot synthesize ribosomes, cannot build proteins, or replicate its DNA if its metabolic network is fractured. A fully biological organism reliant on its own genome could not have survived with these metabolic voids. To reconcile these models, we have to look outside the biological boundary of the cell and into the geochemical architecture of the Hadean Earth. Those gaps were bridged at a specific physical location, the Hadean hydrothermal vent. Early life was not a free-floating sphere drifting in the open ocean. It existed in a state defined as the half-independent cell. These protocells relied on the solid inorganic crust of the Earth to execute chemical reactions their limited genomes could not. Life began as a chemically tethered extension of the Earth's geology. This cross-section of a serpentinizing vent wall shows a microporous mineral matrix. The crust is riddled with tiny, semi-enclosed rock chambers filled with alkaline fluid. At this exact rock-fluid interface, primitive cell-like compartments began to form. Early organic membranes physically coupled to the catalytic mineral surface, creating a continuous hybrid organic-inorganic structure. The geochemical process of serpentinization consumes water. Inside these microfractures, the water-to-rock ratio drops drastically. This low water activity locally increases solute concentrations, solving the dilution problem that would otherwise tear early organic molecules apart in the open ocean. This hybrid architecture functioned as the earliest cellular membrane, trapping chemical intermediates against the vent wall to force continuous reactions. The vents naturally deposit specific transition metals into this matrix: iron, nickel, cobalt, and palladium. These solid-state metals acted as geochemical prosthetics, serving as the functional precursors to biological enzymes. Carbon dioxide interacts with a metallic nickel-iron lattice. Without biology, native metals drive the acetyl-CoA pathway, converting it into pyruvate. Shifting focus, hydrogen gas interacts with nickel. The metal replaces NADH, executing a hydride transfer onto an organic intermediate. These inorganic elements also execute reductive aminations, directly synthesizing simple amino acids. Modern enzymes evolved to be highly specific to single reactions. The native metals are broad spectrum, efficiently catalyzing multiple different reactions across the network. The 400-reaction metabolic core we see in living cells today is essentially a biological mimicry of these vent surface reactions. The enzymes missing from Luca's genome were provided by the vent itself, executing the exact chemistry modern biology claims as its own. Even with the metals providing catalysis, early cellular chemistry faced a strict thermodynamic barrier, generating phosphorylation in water. The half-independent cell completely lacked the complex ATP synthase proteins that modern biology uses to transfer energy. Serpentinized rocks naturally generate phosphite, a compound that is kinetically stable but highly reducing. This equation demonstrates phosphite reacting with palladium and nickel at the vent wall, releasing an electron to activate it. The activated phosphite then directly attaches a phosphate group to an AMP molecule, converting it to ADP and bridging the energy gap. This specific metal-catalyzed reaction provided the continuous, high-energy chemical currency required to drive primordial metabolism forward. The primary pressure test for this geochemical model is availability, specifically the severe crustal rarity of transition metals like palladium. However, the microfractured vent environments concentrate these rare elements locally over vast scales of geological time, bringing them to viable thermodynamic levels. There are also limits to current protein reconstruction databases. Archaeal enzymes are historically undercharacterized, which can artificially inflate the appearance of bacterial-specific inventions. When we compare the two models of Luca, we are looking at two different systems. Gene trees track the transfer of biological information. Structural metabolic mapping tracks the physical origin of catalysis. Luca existed within a complex genetic ecosystem, sharing information and chemical intermediates. But that ecosystem remained physically dependent on the vent's metallic architecture. This timeline matrix tracks the chronological assembly of metabolism. Amino acids emerge first, followed by nucleotides, and finally complex cofactors. But this reveals a paradox. These molecules serve as mandatory reactants for the very pathways that precede their own creation. At the onset of metabolism, these organic cofactors did not yet exist. Biologically synthesized cofactors are evolutionary replacements for the broad-spectrum environmental metals that previously did the work. Internalizing these inorganic reactions into organic cofactors was the critical threshold required to make metabolism self-sustaining. Luca eventually diverged into two distinct lineages, archaea and bacteria. As they split, Both lineages independently invented different enzymes to catalyze the exact same reactions, like alanine dehydrogenase. These independent post-LUCA biological inventions finally plugged the holes in the metabolic network, forever ending their reliance on vent wall chemistry. The hybrid membrane closed off, wrapping the newly synthesized organic cofactors and enzymes into a fully enclosed system. The enclosed cell physically detached from the metallic vent wall. By replacing geological rock with organic cofactors, metabolism achieved true autocatalysis, liberating the first independent cells to leave the hydrothermal vent and colonize the primordial ocean. When we picture the origin of life, we tend to imagine a fully formed, independent cell drifting freely through a vast primordial ocean. For decades, this was the assumed portrait of LUCA, the last universal common ancestor. But a recent study used structural phylogenetics, a method of mapping the physical shapes of modern proteins backward in time, to reconstruct exactly what LUCA was capable of doing. The data revealed a massive hole in that traditional portrait. LUCA was severely incomplete. It completely lacked the crucial enzymes required to synthesize its own basic building blocks, like amino acids. If this ancient organism lacked the internal machinery to build its own parts, there is no physical way it could have survived alone in the open water. To find where LUCA actually lived, we have to look away from the open ocean and down to the deep sea floor, specifically to phosphorylating hydrothermal vents. This animated cross-section shows how LUCA actually survived. Instead of floating freely, this early life form was plugged directly into the rock, essentially outsourcing its missing biological functions to the surrounding geological hardware. Embedded in that rock face were native transition metals like iron and nickel. These metals acted as physical environmental catalysts, pulling in raw elements and forcing chemical reactions to happen, doing the exact same work that modern biological enzymes do today. All of those metal-catalyzed reactions require continuous chemical energy to run. That energy was supplied by phosphite, a highly reactive compound emitted straight out of the vent itself. At this stage in Earth's history, biology as we know it didn't really exist. Life was a hybrid system, permanently tethered to the physical rock that kept it alive. For life to spread across the globe, it had to figure out a way to break that bond and leave the mineral-rich vents behind. To do that, organisms needed to become completely self-sufficient. They had to invent a way to manufacture their own biological catalysts to replace the chemical work that the rocks were doing for them. This diagram demonstrates the solution: cofactors. These are complex organic molecules that early life synthesized to mimic the function of those native transition metals. By producing these cofactors internally, an organism could pack up its metabolism and take it on the go. Researchers can trace exactly when these cofactors were invented by measuring the structural differences between modern protein fossils. This timeline plots early metabolic inventions. Notice this massive gap. Structural data proves cofactors appeared very late, long after basic nucleotide synthesis. LUCA's entire lifespan falls squarely in this early phase, meaning our common ancestor never replaced its rock catalysts. The vital biological tools required to free life from the hydrothermal vents were only invented after LUCA began diverging into distinct species. LUCA appears to have lived and died in total dependence on the vent, making the existence of modern, free-living organisms a biological anomaly. This phylogenetic tree illustrates how our single common ancestor, while still physically stuck to the rock, eventually divided into two distinct branches, Protobacteria and Protoarchaea. The final evolutionary steps required to achieve metabolic autonomy happened completely independently in these two separate groups. Take an enzyme like alanine dehydrogenase. Both branches needed it to perform identical survival tasks, but they each invented completely different, structurally unrelated enzymes to do the exact same job. The same thing happened with their cellular walls. To enclose their newly autonomous metabolisms, both lineages engineered entirely distinct lipid chemistries for their outer membranes.
Only after they finished these parallel construction projects could either lineage finally break its tether to the ocean floor. This means that biology did not achieve its independence once. It happened through two entirely separate parallel evolutionary escapes. This dual escape raises a complex question about how we define the start of biology. Does this mean life essentially began twice? Some scientists point to a concept called remote homology. This is the idea that these distinct enzymes might actually share a common genetic origin, but they mutated so heavily over time that any structural resemblance simply vanished. Others argue for convergent evolution. This theory suggests that because the protobacteria and protoarchaea were facing the exact same harsh environmental pressures, they independently engineered identical outcomes entirely from scratch. Regardless of which theory holds true, our deepest history remains a saga of biology and geology acting as a single unified system, a long geologic tether that eventually fractured, fueling two separate escapes from the rocks that built us.
In modern biology, we define a living organism as an autonomous entity. It is a system neatly separated from its environment by a membrane, powered by its own internal engines. This phylogenetic tree tracks life to its root, the last universal common ancestor, or LUCA. But further into the past, the definition of an organism blurs. LUCA's internal metabolism was incomplete, lacking the biological enzymes to synthesize building blocks like amino acids. It also lacked modern chemical batteries like ATP, meaning it possessed no biological way to power itself. To understand how an incomplete organism survived, we have to look past the biology and focus on the planetary geology that supported it. Ancient serpentinizing hydrothermal vents created warm, microporous rock chambers filled with continuous chemical gradients. These geological systems naturally produce phosphite, a highly reactive molecule that serves as a continuous energy source. When phosphite is exposed to native palladium and nickel in the rock, it triggers a reaction, transferring energy to transform AMP into ADP, which is usable biological energy. This energy conversion happens directly against the rock, occurring without biological enzymes. Early proto-organisms did not generate their own power. Their energy was entirely outsourced to the planet's native geology. Even with an external energy supply, early life lacked the enzymes required to build physical structures like amino acids. Native transition metals in the crust, iron, cobalt, nickel, and palladium, functioned as the catalytic forerunners to biological enzymes. These metals captured raw environmental gases, like hydrogen and carbon dioxide, and forged them into organic matter. These metal-catalyzed geological reactions are chemically identical to the metabolic pathways used by modern cells today. The chemical reactions of the planet were the organism. The rock served as its physical body. Life eventually achieved independence by evolving organic cofactors, like NADH or CoA, to replace the broad-spectrum transition metals found in hydrothermal vents. This internal toolkit made metabolism autocatalytic, allowing the organism to carry its catalytic engines wherever it went. By replacing the planet's metals with organic cofactors, life severed its connection to the rock and achieved biological autonomy. This paradigm changes how we approach the modern search for life elsewhere in the solar system. Icy ocean worlds like Enceladus are prime targets for life that may still be dependent on its planetary geology. Beneath the ice of an alien hydrothermal vent, we might find a protobiological compartment fused directly to the pores of a metallic rock. At this boundary, chemical energy and native metals flow between the planetary crust and the protocell. Our ultimate challenge is determining exactly where the alien ends and its planet begins.